I'll introduce the session. I'll introduce the session, and then we'll turn it over to our first presenter, Ben Nathanson. So this is the Answer Cancer Foundation AnCan's special 1-hour session providing summaries of presentations given by the Prostate Cancer Foundation Scientific Retreat. I believe it was September 2021. And just a spoiler, I don't believe any of us felt that there were any game-changing therapies introduced here. Nevertheless, there were some points of interest scientifically that we'll mention. And we're going to start off right now with Ben Nathanson. So go ahead, Ben.
Okay. Okay. So as you probably know, in ADT, the testosterone is just a game piece. It's the on switch for a molecule in the prostate cells called the androgen receptor. And testosterone is cut off to keep the receptor inactive. And as you also know, cells at some point stop falling for this. Typically, they'll find ways to keep the receptor active without external testosterone. But in 1 man out of 5, the cancer cells do something else. They change their identity and become a kind of cell that doesn't have an androgen receptor. It's as if cells in your kidney turned into liver cells. The cell type these cells become is called neuroendocrine. Neuroendocrine cells aren't scary when they're in the organs they belong in, but these are cancer cells, and neuroendocrine prostate cancer is a dead in 2 years diagnosis. PCF session 1 was devoted to this particular evasion, and I'm reporting only on one of the papers, Amina Zubeidi's talk. Her team has found a way to target a known driver. It's been known for a few years in neuroendocrine prostate cancer. It's a molecule that the androgen receptor normally suppresses. It's spelled BRN2 and it's pronounced brain2. They developed a brain2 inhibitor that plugs up the part of brain2 that reads DNA. And they show that this inhibitor completely blocks the action of brain2. The effect was the same as if they destroy the gene that produces brain2. And as you see here, the inhibitor stops neuroendocrine cells from growing. Just to go through this a little. So these are cell lines. I guess you cannot see what you can't see my cursor. So you see Marcus. Oh, good.
You can see your cursor.
Okay, good. So these are cell lines for different kinds of prostate cancer. This top one is castrate resistant, but it's not neuroendocrine. This one is enzalutamide resistant, but it has neuroendocrine markers. And this one at the bottom is sheer neuroendocrine. And you see that as more brain2 inhibitor is added, growth goes down, in this case, to 0. I don't know if I just said this, in mice, the inhibitor was as effective as carboplatin without the toxicity. So this is pretty exciting. The inhibitor has no name yet except for its lab name, which is B18-94. Dr. Zubeidi did not talk about what her next step would be. Okay, where's Jake Hannam, the mute king, to...
All right, I can take care of it.
Thank you. Okay. The subject of this talk, oh shit. The subject here is artificial intelligence software cleared by the FDA that reads and interprets PSMA scans. The AI was developed The AI was developed by Lantheus, the company that developed the Pylarify scan. So you may be thinking, AI, how exciting. Turns out that commercial use of AI in medicine is not so new anymore. The FDA has been approving AI for, as you see, several years. And what's more, this is one of several AIs in prostate cancer. Here is another one approved somewhat later to help with Gleason grading. There are at least 3 to help grade MRIs, and there's another to help identify focal prostate cancer for laser ablation. But novel or not, this software addresses a real need. You don't have to be a doofus to be misled by a PSMA scan. PSMA uptake can be tissue dependent, so medium dim can mean the same as bright depending on organ. There's also variability from patient to patient. So one guy's medium dim is another guy's bright in the same organ with the same mets. Most of us already know that the PSMA lights up organs that are unrelated to prostate cancer. But in addition, in the second slide, or the second quote, other diseases can light up the scan, even things that are not obviously false positives, but they're entirely different diseases. Sorry. As a result, complex guidelines have been developed for grading scans, one of which is called PROMIS for, as you see, prostate cancer molecular imaging standard evaluation. And as you can see, because there are so many things to trip you up in a PSMA scan, the classification has a large number of criteria. And anything with that many criteria is going to be tedious to do, time consuming, and is likely to have inter-observer variability. Every time you consider one of these criteria is another opportunity to have your judgment different from someone else's. So this software called PROMIS is designed to automate this assessment. As a first step, it identifies the body structures in the CAT scan, individual bones and individual organs. It uses that organ information, bone and organ information, to apply the PROMIS criteria and indicate what radiologists commonly call regions of interest. You can see, though, it's not quite as clear as it might be in this slide, that the AI version is much more definitive on regions of interest than the non-AI version. It has also taken stuff that lights up that's not interesting and has not marked it as a region of interest. So you might wonder, if PMA scans are so deceptive, how Lantheus can claim their AI is correct, or when you get right down to it, how any reading can be trusted. It turns out that first, experienced observers tend to reach the same conclusions. And second, that these consensus conclusions are correct because they can be validated by biopsies. So the Osprey trial was what verified Pylarify, and they took the Pylarify patients and used them to develop a PROMIS, and found that they did get segmentation accuracy, meaning it did define the tissues and organs correctly. It got consistency and quantitative assessment. That's another benefit of something like this AI, and in principle, in a grading scheme like PROMIS, that you get a number output on severity, and that it had high sensitivity. But we have to bear in mind that this correctness is only correct insofar as false positives. None of this helps with false negatives, which I will talk about in a minute. So I meant to ask the speaker about the business model. It would be nice if Lantheus gave the software away with the Pylarify tracer like Cracker Jacks, but I suspect it's sold separately. So smaller hospitals that are most in need of this software might be the places least likely to be using it. The research that to me perhaps had the greatest medium-term impact was this poster paper from Robert Flavel at UCSF. This image and the next are borrowed from another talk by Michael Hoffman. When PSMA is good, it's very, very good. Wouldn't we like to be one of these lucky guys whose Mets disappeared when they got lutetium? But when PSMA is bad, it's very, very bad. Here's a standard FDG scan finding lesions that PSMA didn't. How big a problem is this? We hear different numbers. At a minimum, PSMA is unlikely to help men with treatment-emergent neuroendocrine prostate cancer, which is 20% of the castrate-resistant population. So on the diagnostic side, there are tumors that are missed, and on the treatment side, is collateral damage to the organs that are also in the PSMA club. At UCSF, they found a theranostic combination that appears to avoid both of these shortcomings. It works for all kinds of prostate cancer, and it works even better if it's neuroendocrine. This slide shows higher tracer uptake for this theranostic combination compared to PSMA, and that this uptake includes the treatment-emergent neuroendocrine prostate cancer, which the PSMA barely registers. The theranostic combination they've discovered is also almost perfect in targeting only prostate tissue and cancer. The only other tissue that's avid is placenta. That's biology for you. The biomarker is CD46. It's a membrane protein like PSMA. CD46 is an unlikely biomarker because it's expressed literally everywhere in the body. The only cells that don't express CD46 are red blood cells. What the UCF team has discovered is that prostate cancer seems to change CD46 uniquely in a way that makes it different from all the other CD46 in the body. Exploiting this, they've developed an antibody, YS5, that will only latch on to cells that have this alteration. The antibody isn't for immunotherapy. It carries theranostic payloads, and those payloads are what Flavel and his colleagues have developed and tested. For PET, here we go. For PET scans, they've tied the positron emitter zirconium 89 to the body, and for therapy, they've tied the alpha emitter actinium 225. And both of these conjugates have been shown effective in mice. Both are planned for trials. And a little advertisement. You can be the prostate cancer patient who greenlights funding for projects like this. You can contact me offline for details about the PCRP program. So although this is not mentioned in Flavel's paper, it's worth mentioning an ongoing phase 1 trial that uses YS5. And the trial YS5 is attached not to a radioactive material, but to a chemotherapy drug. It's still in phase 1. It's too soon for results, but it was reported on recently, and there's early word on side effects. There's unfortunately some neuropathy even with YS5, but it may not be severe. One thing you can say about PSMA is that you can't yet say about CD46 is that it's been studied by hundreds of researchers over dozens of years. PSMA sprang into the news like it had just been discovered, but it was discovered in the 80s, and its theranostic potential became clear in the 90s. The CD46 epitope we're talking about here was discovered in 2010. In drug development years, that counts as infancy. So its luck might still run out. The obvious question is, if CD46 is such a big deal, why did PCF make it only a poster paper? And I don't think it's that they were underwhelmed. I think it's that this work has been ongoing, and it was talked about in last year's retreat. This is an incremental step in which they've added the actinium-225 to the mix. And that's my talk. I can answer questions.
Say that was a terrific job. Excellent job.
Thank you.
And really good. For those of you wondering about the term theranostic, that's the term that we use around radionuclides. Just some of us know that, some of us don't. And radionuclides are the things like lutetium-177 and actinium. There were just a number of really great papers that you brought to light. I'll turn it over to Dr. John. Go ahead, Dr. John.
I need technical support. I don't know how to turn off my screen sharing, or have I already turned it off?
At the bottom, you should be able to.
Yes.
Oh, I see. It's not in my one sec. At the bottom of the main
screen.
The screen with the faces?
There should be at the bottom of your screen, there should be the screen logo.
No, at the
wherever you see your microphone and your camera.
I don't see it. The window has changed because I'm displaying.
Okay, so minimize. We'll take you off being presenter. Let me do that, and that'll make it easy.
If that did not work, I can just log off.
No, you don't have to do that. We'll figure this out. We'll make somebody else. We'll make me the presenter, and we'll make Len the presenter for the time being. John, let's not ask you a question whilst we're figuring this out.
What did you mean when you said that one of us could be the person who activates?
Oh. So this Defense Department program is actually for civilians as well. It's congressionally funded, and the mandate. Take a step. This is the second largest funder of prostate research in the country after the National Institutes of Health. It is larger than PCF. It is larger than anybody else that's not in the government. And every bit of research that they fund, every review panel includes a consumer reviewer. And you can elect to be one of those consumer reviewers. You have to be nominated and you have to pass an interview. But you can actually be up close and personal with studies like this as they happen. So that's what I meant, Stephen.
Thank you. Forget that I was muted.
Yeah, and just one other thing I want to add from Ben's talk. Steve asked in the chat window, is this specific to Pylarify? It is specific to Pylarify. They've developed this to accompany the Pylarify scan, and we've been talking about it, as some of you I'm sure know, many times in the past 3 or 4 months, that there is now AI available to help community centers interpret. A good example of somebody who did not do that well in terms of getting his scan read was Joe Blanchette. He really went through the ringer trying to get somebody to read his PSMA scan accurately.
Yeah, Rick, Joel here. I talked to my radiation oncologist today, and he said that he gave me 2 MRIs a week ago. And he asked for a very, very sensitive MRI, which meant I was in the machine for 2 hours. So
hold on a minute. Hold on a minute, Joel. Herb, we got to mute you.
I've got trouble.
And
Troubles.
Joel, we don't have time to follow up with this now. Let's follow up with it in the session that follows, if we may.
Okay.
Hey, I have a
Len?
quick question.
Go ahead, Dennis.
Yeah, thanks, Ben. That was a great presentation. Question on neuroendocrine. How is that diagnosed?
Yes, good question. They can do it by, usually in a very small percentage, in most cases it emerges under treatment. They can biopsy the tumors to find it. They have been developing, you know, since biopsying tumors is either difficult or sometimes impossible, they are developing, but it's not available yet. They are developing liquid biopsies that can find it. That's a good question.
Len?
Yes, I think I'm going to be the next presenter. If you could do that for me, Jake, or someone.
I believe I did, Len.
Okay.
Sorry, I thought you said it was going to be John next.
All right. Can everyone see my screen?
Yes.
Okay. So the first presentation I'll be summarizing was on the therapy trial, which, whoops, hold on, which was a presentation by Michael Hoffman. Ben made a brief passing mention of him. He is with the Prostate Cancer Theranostics and Imaging Center for Excellence, or PROSTIC, in Australia. And they did a phase 2 trial with lutetium versus cabazitaxel. We're more familiar with the trial, the VISION trial, which was lutetium versus best standard of care, which was basically the physician's choice of a second-line anti-androgen after the first one had failed. That approach had been criticized by Vinay Prasad as not really being an active control, because as we know, when you fail one second-line anti-androgen, the response rate to adding a second is pretty low. So these guys used a more active control, namely cabazitaxel. So this was in MC. By the way, let me introduce terms, make sure we're all on the same page. MCRPC, metastatic castrate-resistant prostate cancer. So this was a trial in patients with MCRPC who had progressed on second-line anti-androgens and docetaxel. It was a phase 2 trial. Lutetium, as most of us know, is a beta emitter with an approximate 1 millimeter path length, which limits off-target damage. Now, a millimeter, if you take a pencil and you make a mark on a page, the pencil point is about 1 millimeter. So, but within that 1 millimeter, there could be 1,000 cancer cells. So lutetium has a half-life of 7 days. So even after lutetium is taken up by the cells, it continues to kill adjacent cells for days afterward. SUV is another term we should define here, standard uptake value. that is another way to talk about that is the avidity of the PSMA stain, of the intensity of the stain. And it should be, for this trial, there had to be at least one lesion that had an SUV of 20 or higher, and that's compared to background radiation of 1. Primary endpoint was decline in PSA of 50% or more. Secondary endpoint, delay in radiographic progression-free survival measured by either CT or bone scan. Okay, so here's another major difference between the therapy trial and the vision trial. In the therapy trial from Australia again, all patients were mapped with both PSMA PET scan, gallium PSMA, and an FDG PET scan. So what's the advantage of this? If a patient had positive lesions with the FDG scan that didn't show up on the PSMA scan, that's an indication that there are non-PSMA active lesions. And so those patients were excluded, as you can see here, 28% of patients were excluded due to what they call discordance between the two scans. And Ben had a similar slide to this. So on the far left, you have PSMA PET scan, and you see a bunch of lesions. On the far right, you see an FDG PET scan, and there are even more lesions. And in the middle, you see them superimposed on each other such that the reddish lesions showed up with FDG, and the blue lesions showed up with PSMA. So you see, this patient would have been excluded from the trial. And why do we want to do that? We want to make sure that the patients we select have a good chance of responding to the therapy. So the primary endpoint was reduction of PSA by at least 50%, and 2/3 of the men assigned to lutetium had a PSA decline of 50% or more, compared to only 37% with cabazitaxel. Secondary endpoint was a delay in the time to progression. And although the PSMA, lutetium PSMA, they were 37% less likely to progress than cabazitaxel, very good. However, this was a surprise to me. I don't know if it'll surprise anyone else. At 12 months, only 1 in 5 men who were assigned to the lutetium arm had still not progressed. So, you know, this is a great new therapy, but I think there's been a lot of hype surrounding it. And so what we see here is that you can expect that only 1 in 5 men will still be responding positively to lutetium after just 1 year. The objective response rate, the ORR, is a measure of tumor shrinkage on the CT scan. That occurred in 49% that were assigned to lutetium, compared to 24% for cabazitaxel. Now you see I made a note here for myself to see slide 5 for a comparison to the vision trial. And I can give you a spoiler alert right now. The 49% who responded here, who had tumor shrinkage with this trial, that number drops down to only 29% in the vision trial. And why is that? Because the vision trial did not exclude folks who had non-PSMA active lesions. So no surprise here, lutetium had resulted in fewer side effects than patients with cabazitaxel. Severe adverse effects, which are grade 3, 4 toxicities, reported by physicians, and there were 33% with lutetium compared to 54% with cabazitaxel. And patients on lutetium experienced less symptoms in multiple, less toxic symptoms in multiple domains, including hair loss, diarrhea, fatigue, insomnia, dizziness, urinary symptoms, sore hands and feet, and skin rash. Now, so the Novartis trial, as we already discussed, was evaluating lutetium versus best standard of care in, again, progressive metastatic castrate-resistant prostate cancer who pre-tested positive for PSMA, but they did not exclude patients who were FDG positive because they didn't include an FDG scan. FDG, by the way, for those who don't know, is fluorodeoxyglucose. It is a radioactive marker for glucose uptake. And most cancers have a greater uptake of glucose because they're metabolizing faster, they're growing faster. Okay, I already mentioned this. Patients must have progressed through hormonal therapy, and which means they were, hormone resistant, and yet in the control arm, they were assigned another hormonal treatment. So obviously that was not going to be a very active control arm. So here we have improved overall survival, 38% reduction in risk of death in the lutetium arm, and 60% risk reduction for rPFS. And that's, again, compared to a non-active control arm. We've already covered the toxicities for lutetium in the other trial. But you can see here, they're really not that bad. They're manageable. Thrombocytopenia, for instance, that's low platelet count. Lymphopenia, low lymphocyte count. Fatigue, anemia, 12.9%. So here is what I already referred to. The objective response rate here was only 29.8% for the lutetium arm. We remember that it was 49% in the therapy trial. So it's important to properly select patients for trials with lutetium. Okay, the PRINCE trial. PRINCE stands for PSMA, lutetium radionuclide, and immunotherapy for prostate cancer. The only thing I want to, this is a busy slide here. So they're combining pembrolizumab with lutetium. So why are they doing that? Well, we've already seen that they're saying here recrudescence of disease underpins the need for novel treatment. So this means recurrence of disease. And we've already seen that even with successful lutetium treatment, patients regress or progress. And so the hope is that pembrolizumab can elongate the response rate with lutetium. Pembro by itself, as we know very well, has very little anti-tumor activity in mCRPC. So the hope is that due to the radiation from lutetium can potentially induce immunogenic cell death that may be enhanced by immune checkpoint inhibition. This is just the trial design here. We don't need to go into that for interest of time. So the conclusions were that lutetium and pembrolizumab has promising activity with a PSA response rate of 73%. Note, so PSA with a decline of greater than 50%. 73% of patients achieved that response rate. And radiographic progression-free survival at 24 weeks was pretty high, 65%. Now, unfortunately, they don't have data going out 1 year. If you recall, the rPFS with lutetium alone, without pembro, was down to 19% at 1 year. So, they're still following the patients in this trial. Also good news here, toxicities were consistent with those of single agent lutetium and single agent pembro. So there's no synergistic toxicity. And they're going to be studying biomarkers to see who would be the proper patient to select for this combination. Okay, this I'm going to zip through quickly also because the Lumiere trial is run by Clovis, and it's lutetium, not lutetium PSMA, but lutetium FAP. FAP, this is also a theranostic. Gallium-68 FAP is the diagnostic portion, and the therapeutic portion is lutetium FAP. FAP standing for fibroblast activation protein, which can be highly expressed in certain cancer cells. And so basically, they're targeting the stromal cells, which are the supporting cells for the cancer cells, helps them grow and spread. And FAP is expressed by the stromal cells, and that's what's being targeted. And of course, lutetium will kill those cells, and being that they're right on top of the cancer cells, it'll kill the cancer cells as well. The bad news is that only a small percentage of prostate cancer stromal cells actually express FAP. This trial is being run by Dr. Thomas Hope, who is the principal investigator at UCSF, and he's investigating the FAP theranostics. So key questions are, what are the patient demographics and clinical factors associated with high expression of FAP? These are questions that need to be answered. What's the overlap of PSMA and FAP expression in prostate cancer? And if anyone's interested, they are recruiting patients in this trial. The last thing I'm going to cover here is insights and predictions by Dr. Neil Bander. He's a professor of urologic oncology at Weill Cornell. He showed a table comparing the properties of alpha and beta emitters. Alpha emitter, such as actinium-225, Carl Foreman was in a trial with actinium. Other guys were in trials with the beta emitter, lutetium. So the important things to look at here is simply that the maximum range in tissue penetration with the alpha emitters is very small, 50, just say a unit of 50. And with the beta emitters, it's much, much greater. So there's a broader penetration of cells, 1,700 compared to 50. However, look at the linear energy transfer. This is you can look at it as the power of the radiation being emitted. So obviously here, it looks like the alpha emitters are about 500 times the energy of the beta emitters. The alpha emitters cause double-strand breaks. Beta emitters, that's DNA, double-strand breaks, whereas the beta emitters cause DNA single-strand breaks. So you only need one hit of an alpha emitter to kill a cell, whereas you need 1,000 hits of a beta emitter. So anyway, his prediction is that dual targeting using both alpha and beta emitters will prove to be beneficial. And he says that we'll see dual targeted PSMA therapeutics delivering increased potency and clinical benefit. All PSMA radiopharmaceuticals will be approved for use in earlier stages of disease. And they will have the potential to cure in these earlier settings. These are just predictions. It's not based on any data. Oh, I forgot I had the SureCap. Okay, so just very briefly, SureCap is a study being conducted by Charles Ryan, who is now with the Prostate Cancer Foundation, and it stands for Survivorship Research in Prostate Cancer. Because prostate cancer patients are living longer, there's no research on the consequences there's potential for competing health risks: cardiovascular disease, sarcopenia, which is muscle weakness, and falls, consequences of long-term androgen deprivation therapy. You know, he's going to be studying this, and he's going to be studying cognitive decline. This was a bit disconcerting to me, as I'm sure it will to anybody on ADT. So we see that compared to healthy controls, there's a 40% gap in patients on ADT with regard to immediate span of attention, 50% gap with regard to visuospatial activity, and 40% gap with regard to executive function. Okay, so what's going on here? ARACOG is a study which stands for androgen receptor and cognitive function in darolutamide and enzalutamide patients. They will soon begin recruitment, and it will be randomized to avoid patient selection bias. So the big picture, what's going to be the outcome of this study? Improved outcomes for prostate cancer patients. We learned something about the role of the androgen receptor in cognition. We enable or validate cognitive evaluation in oncology clinics, and we develop genomic guided survivorship therapy decisions. I'm not going to cover this because this is not terribly important. We already know the potential benefits of a direct agonist versus a direct antagonist versus an agonist in ADT drugs. So I'm probably going to have the same problem that Ben had in closing out my screen here.
I got it, Herb. I got it, Len.
Okay, thanks.
Hang on. Here we go.
All right.
Should be able to go, Herb.
Okay, go ahead, Herb. Unless we have questions.
He was having some issues earlier. There we go.
Okay. Now,
Hold
screen.
on a minute.
Okay.
Hold on a minute. How are we doing for time, Len? I mean, I know we've got Herb, we've got Jimmy on exercise. I mean, how are we? And we've only got 15 minutes left. So how do you want to handle this?
Yeah, so we're gonna have a second session in that case. I wasn't even aware of Jimmy. Somehow, I guess we didn't include him in the emails that went out about posting presentations on the Google Docs shared drive. So, you know, he didn't even show up there. Sorry about that, Jimmy. I don't know how that happened, but Herb, it's 7:45. Do we have time for your presentation?
I'm going to do as much as I can, and we'll see what we get, where we get.
Well,
okay. And whoever we don't get to, we'll get to in 2 weeks.
Right. If I have to interrupt and save something, I'll save something. Okay, so my take on this meeting was I was really focused on the science rather than the clinical, because this wasn't a clinical meeting. And so we all know about olaparib and how it's been said that it's good for BRCA1 and BRCA2 mutations. But here, these people, this is a study that showed that in fact, if you have a mutation in PALB2 or BARD1, and if you take a look at this image in the upper left-hand corner, they're both molecules that act in combination with BRCA1 and BRCA2 in homologous recombination. And they're similar, therefore, when you have a PALB1 or BARD1 loss, you get homologous recombination deficiency. And this is just comparing an image. I don't know if you can see my pointer, but on the upper right, you can see the black bar is normal. And when they knock down BRCA2, things are down. They knock down PALB2, things are down. They knock down BARD1, things are down. And in the lower right-hand corner, what you can see are survival curves. And here they give you the sensitivity to olaparib, which we all know is good for homologous recombination in the case of BRCA defects. But here, this is showing when you knock down PALB2 or you knock down BARD1 in these models, that olaparib then becomes a viable drug to preserve life. So this adds 2 more gene knockouts, PALB2 or gene mutations. If you're screening for gene mutations, like with a foundation medicine screen, if you think of PALB1 or BARD1 loss, that could allow you to be eligible for olaparib. And then this is the study that Len mentioned, basically relugolix versus luprolide, and what it basically says that there's no big difference here. If you look at the survival, these Kaplan-Meier curves of people taking either relugolix or luprolide, you don't see any big difference. So while the side effect profile may be different between the 2 drugs, the antagonist versus the GnRH drug, there's no real difference in biological outcome. So again, that becomes a matter of then preference in terms of what you think is going to be the appropriate drug for you. Okay, here's yet another potential drug targeting, and there are a fair number of, and this is from Felix Fang and a bunch of collaborators. There are a bunch of, as I mentioned with PALB1, there are mutations in the tumor that allow you the potential therapeutic approaches. And here's a strategy for CDK12 deficient cells. And here's just an example. CDK12 is a transcriptional cyclin-dependent kinase which phosphorylates RNA polymerase II, and that's what makes RNA here from DNA to regulate transcription elongation. And what you can see here in the upper right, and then here showing that in fact people with CDK mutations are enriched in cancers with poor clinical features. So that's again an indication that oops, sorry about that. Let me go back. That CDK is a potential treatment. And then here, this is in a mouse xenograft experiment with CDK12 knockout cells, and they're just a bunch of pictures, but it basically showed that, in fact, there is some efficacy of rucaparib in a CDK12 mutation. And then here they looked at a subset of patients in the Triton-2 study, showing that yes, they had a PSA decline. Of course, it may not be permanent, but again, pointing to the fact that the more we learn about some of the basic biological actions due to mutations in these pathways for either DNA synthesis or RNA synthesis, that we can point to treatment options. Now, we heard Ben talk about neuroendocrine prostate cancers. And it turns out that if you look at neuroendocrine prostate cancer samples, there is a protein on the surface called DLL3, which is on the, here's a sort of a molecular diagram of DLLC3 staining on a surface of a cell, and basically is an atypical pathway that is overexpressed on other tumors on neuroendocrine tumors. And then a study of 423 patients with prostate cancer, DLL3 was expressed in 77% in neuroendocrine prostate cancer samples, and then with 64% of the cells expressing DLL3. And so here's just another way of showing that on the right, that basically neuroendocrine prostate cancers express this protein called DLL3. Okay, what does that mean? It means maybe it's a potential target. And then here they show that DLL3, as you can expect, if it's neuroendocrine, it's inversely correlated with PSMA and STEAP1. And if you take a look at the lower left-hand corner, it's pretty bad if you have DLL3 positive tumors versus not. I mean, the survival rate is 20, basically, roughly 3 months versus 22 months. So it's pretty bad. So they now use it, they're making a BiTE. They're making a BiTE molecule, which is just like a lot of other BiTEs, which is designed to use BiTE technology to attack DLL3 neuroendocrine cells, which would be, we've all heard of BiTEs for PSMA. So here's an example of the neuroendocrine phenotype, which is pretty severe with a potential target. And there is, they have a clinical trial with small cell lung cancer, and potentially it might be effective in prostate cancer as well. So I thought that because neuroendocrine phenotype is so bad, we need more approaches to neuroendocrine type. And then here is survival in a model after BiTE treatment. And again, if you look at the black line, it's control in an animal model, and in this tarlatamab, which is the BiTE towards DLL3, it increases survival. So I think we can look for a clinical trial with this antibody in DLL3 positive prostate cancer, NEPC. And then here's here's the criteria, and then histological, and so it's available. One thing to note, and that was brought up here, is that anti-drug antibodies, that basically when you give people BiTEs, they're giving them a protein that's foreign to the body. So it could generate an immune response. So we have to worry with all the BiTE trials about potential anti-drug antibodies with BiTEs. Again, here's another pathway. The adenosine
I think we need to stop at this point, Herb.
Okay. I can continue later.
Did anybody? I had a question for you.
Yes.
So PALB1 and PALB2, I thought it was right, and Len's just confirmed it, that when they approved olaparib, they approved it for a whole bunch of mutations that included PALB1 and PALB2 and BARD1. But in fact, there was no evidence that people that expressed PALB2 or BARD1 responded to olaparib. And in fact, nor CDK12 for that matter. The only thing they seemed to respond to was BRCA1, BRCA2, and
I've seen that too.
Possibly ATM. There was some limited response in ATM. So, we're talking about getting people excited if it shows if PALB1, PALB2, BARD1. But Len and I specifically went back to Foundation Medicine. We said you can't call these drugs, these mutations actionable because the evidence isn't there.
Okay.
And Len, do you want to comment on that? Because you did a lot of research on that at the time.
Yeah. So what they did in that trial is they had one arm that had BRCA1 and 2 and ATM, and another arm that had about a dozen other genes in the homologous recombination pathway. And so they showed that in in arm 1, let's call it, which was BRCA and ATM, there was a good response. Now, they didn't provide arm 2 specific results. Instead, they combined the results of arm 1 and arm 2. And what happened was, when you combined them, it still looked like olaparib was helpful, but only because arm 1 was boosting the response of the combinations of both arms. So it was kind of a fake out, and a lot of oncologists realized that eventually they realized that this was a very poor study, the way the data was analyzed, and that there was no real evidence that anything other than BRCA1, 2, and ATM were actually actionable with olaparib.
Right, and my question would be on those graphs that you showed with BARD1 and PALB2. We don't really have time to get into it. Were those in mouse trials or were those in? So
yeah,
I don't know whether. So guys, don't get excited if you see PALB2 or BARD1.
right. Okay.
And you think, I'm good for olaparib. Not so sure. Okay, hold your horses is what I want to say. But so and so we're very concerned that we show you this research and then but people take it in context, and I'm just trying to place it in context here.
Mm-hmm.
We are moving in. It's now 6 o'clock my time, so 8 o'clock Eastern. And Len, that was terrific, but as I suspected, we're going to have to continue this. We're going to continue it before the next 8 o'clock session. We'll come back to Oliver Sartor, who you see in the top right-hand corner there. And we'll set up everybody else. But what we're going to go to right now is a regular session of our high-risk meeting. Now,